Biomix-Up. Today in this video we are going to discuss about the prokaryotic DNA replication. In the last two videos we have already discussed about the basic of DNA replication, what are the enzymes required in the DNA replication, and about the DNA polymerase 1, 2 and 3. So in this video we are going to discuss about the prokaryotic DNA replication. So the prokaryotic DNA replication takes place in the cytoplasm. Okay, and this site and the prokaryotic system is having only a single origin of replication. This is the site from where the replication starts, and this process takes place bidirectionally. That means from two sides the replication takes place. Here there is a creation of a bubble, and from this bubble, from this side DNA replication takes place, and from this side also DNA replication takes place, and it forms two copies. Okay, from a single cell two copies can be formed by the process of DNA replication. We can see here that this is a fully methylated origin. Okay, now let us discuss what is methylated origin and what is hemimethylated origin. This is the DNA strand. This is the DNA strand which is the parent DNA. This is the parent DNA. This parent DNA is having fully methylated origin in the sequence GATC. GATC is the sequence in which the methylation takes place in the adenine. Okay, in the adenine the methylation takes place. What is hemimethylation? When there is a copy of the DNA or the daughter strand, initially it is not having the methylation, so it is so from that reason there cannot be any DNA replication take place. Okay, suppose this is a DNA. This is a DNA which is a parent DNA. This is the parent DNA, and this parent DNA is having methylation. It is having methylation so that the DNA polymerase can come and bind, and it will help in the process of replication. But this is the new strand. This is the new strand. This is a new strand. It is not methylated. It is hemimethylated. Like some portions are methylated. Okay, but a whole DNA is not replicated. So from here the DNA replication cannot take place by the help of the enzyme. So this is the basic difference between the hemimethylation and methylation. So here are certain proteins or we can tell that enzymes which are present in prokaryotes and eukaryotes. In case of prokaryotes, DNA polymerase 1, 2, 3 mainly works. Okay, so this we will discuss later about the eukaryotic DNA replication. Now let us see here the hemimethylation as well as the methylation inside, how they are being regulated. So this is the red part which is present. It is the methylation part which is present in the adenine of GATC sequence. We can see here that there is a GATC sequence, continuous GATC sequence in the DNA, and there is methylation in the adenine. This is the parent DNA. Okay, this is the parent DNA. After the DNA replication, after forming a copy of this parent DNA, this is a daughter strand, and here there is no methylation. So who is preventing this methylation? There is a protein which is known as SEC-A. SEC-A binds in this GATC sequence of the new strand. Okay, the SEC-A protein. SEC-A is a protein. SEC-A is a protein which binds in the new strand in the GATC sequence, and it does not allow the enzyme which is responsible for causing methylation. The enzyme is DNA methylase. The SEC-A protein, it inhibits the DNA methylase from binding in the GATC sequence for methylation. That is the reason as a SEC-A protein, here we can see this is a SEC-A protein which is binding in the DNA sequence, GATC sequence. So here the DNA A, which is the initiator protein, this DNA A, it is the initiator protein who is responsible for initiating the DNA replication. This DNA A protein cannot bind in this region. Okay, after time passes, this DNA methylation, when the SEC-A protein is removed after some times, after 20 minutes this SEC-A protein it removes, then the DNA methylase comes and bind in this region, and in the daughter strand there is methylation in the A sequence, adenine region. Now this daughter strand is also now ready for the DNA replication. So the initiator protein which is known as DNA A comes and bind with the elimination of the SEC-A protein. Here we can see that here the SEC-A protein is removed. Only one SEC-A protein is removed. Okay, here only one SEC-A protein is removed. So methylation is taking place one by one. Only methylation is taking place. DNA methylase is coming and binding in this region. One by one it is binding, and the DNA A is binding continuously, and the methylation is and the DNA replication. Let us see how this DNA replication is taking place in case of prokaryotes. In case of prokaryotes, there are certain sequences which are present. They are known as the 9-mer sequences and 13-mer sequences. Here we can see that here there is 9-mer sequences. 1, 2, 3, 4, 5. 5 9-mer sequences are present and 3 13-mer sequences are present. Okay. So this DNA replication, when it started, it binds, first comes and bind in the 9-mer sequence. The origin is taking place, or we can tell that the initiator protein is coming and binding in the 9-mer sequence. Then the unwinding of the DNA by the helicase is taking place in the 13-mer sequence. So why it is so? It is taking place in the 13-mer sequence. So this 13-mer sequence is also having some of the sequences like adenine base pairs with thymine by the help of 2 hydrogen bond, but guanine base pairs with cytosine by 3 hydrogen bonds. So it is easy to break this adenine and thymine bond because it is having 2 hydrogen bonds. So in this region, in the 13-mer region, that they are rich in adenine and thymine. So it is easy to break these bonds. So the initiator protein comes and binds in the 9-mer sequence, and the DNA helicase works or the unwinding starts in the 13-mer sequence. So who does this? There are certain enzymes like DNA helicase is doing the end. DNA helicase is unwinding the DNA in the 13-mer sequence. Here we can see in the 13-mer sequence it is AT rich, and DNA B is a helicase which is unwinding this DNA. Okay. Now the DNA B is a helicase, and there are certain enzymes which are required for this DNA helicase. After the process of DNA helicase, there are certain enzymes which are required like single stranded binding protein. Why the single stranded binding protein is required? Because when the DNA is now in a single stranded form after the helicase work, to prevent from again recoiling, the DNA single stranded binding protein binds in the single stranded DNA. The next is DNA C. Okay, what is DNA C? It is a sliding clamp. DNA C which is present, it is a sliding clamp which binds in this DNA. So what is the requirement of the sliding clamp? Because it is helping the DNA B or the helicase to do its work properly or to maintain its work properly. Okay, let us see by this picture. Here there is a protein which is known as DNA A, which is an initiator protein. This DNA A is an initiator protein which binds in the ORIC site. Okay, which binds in the ORIC site, and in case of prokaryotes it is only having a single ORIC site. Okay, it is having single replicon site. So here the DNA A protein comes and bind in the 9-mer sequence, and the helicase work or the unwinding starts from the 13-mer sequence. There is another one enzyme which is known as DNA B, which is helping in the unwinding of this DNA, which is a helicase. So the helicase needs to be loaded. It is being done by DNA C, which is the helicase loader. This DNA C which was present, it is a helicase loader. DNA C is the helicase loader. So after that, another one enzyme is required, which is known as primase, or it is known as primase G. So what is the work of this primase G? As we have already discussed in the earlier videos, so DNA polymerase does not have the ability that it can initiate the work of DNA replication. So it requires a 3' OH group. But this primase enzyme which is present, it is a RNA dependent primase enzyme which is present. Primase enzyme which is present, it is a DNA dependent. It is a DNA dependent RNA polymerase. So it has the ability that it can, by seeing the DNA, it can produce a short strand of DNA which is having a 3' OH group. Then here from this reason, the primase enzyme is removed, and the DNA polymerase comes and bind in this region, which will help for further elongation or further replication of the parent strand to make a copy of itself. Okay, so here we can see here the sliding clamp and the helicase sliding clamp and the DNA polymerase both are winding and they are synthesizing the DNA. And the DNA replication, as it is bidirectional, in the two directional both the replication takes place, and there are multiple DNA polymerase working. In the leading strand there are only single polymerases for winding, and in the lagging strand there are 2 DNA polymerase binding. So here we can see that here there is a continuous stretch of DNA from 5 prime to 3 prime. When the DNA polymerase is synthesized in 5 prime to 3 prime direction, it is continuously synthesized. It is known as leading strand. When it is synthesized in the lagging strand, it is when it is synthesized by making some of the fragments, it is known as lagging strand. Okay, this was all about how the DNA replication takes place in case of prokaryotes. By the help of other diagram also we can understand how this process is taking place. Here there is a enzyme which is known as DNA B. DNA B was a helicase, DNA G was a primase. This primase was responsible for making a short strand of DNA which will be having 3' OH group, so that the DNA polymerase can come and bind and continue the elongation process. Here there is a leading strand. Here there is 5 prime to 3 prime. As it is continuously synthesized, it is known as leading strand. And in the leading strand, there is no bend. Okay, so the DNA replication is continuously synthesized. But in case of lagging strand, there is a short pair of this DNA. Short patches of this DNA. So there are in between, there are also primase enzymes which are present. This primase enzymes, or we can tell that primase, so short strand of DNA which needs to be removed. That is being removed by DNA polymerase 1. Okay, how it will be removed by DNA polymerase 1? Like suppose, see this. Suppose this is a parent strand, this is a parent strand, and it is having short strand of DNA. And these are the primase enzymes. So here the DNA polymerase 1 will come and bind here, and it will cut this DNA. And this DNA polymerase 1, as it is having low processivity, it will fall off. During this time, the DNA polymerase 3 will come and it will continue the synthesis of this DNA, and it will fill the gap. In this way, DNA polymerase 1 and DNA polymerase 3 help in sealing the gap which is done by the lagging strand. Or we can tell, due to the primers, there are certain places where this primase needs to be removed, and there will be filling of this DNT base. So this work is done by DNA polymerase 1 and DNA polymerase 3. This was all about how the DNA replication is taking place in case of prokaryotes. In the next video, we will discuss about how the eukaryotic replication taking place. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.